At this point, we've looked at the mechanical side of muscle contraction. We looked at the muscle fibers, the myofibrils, the sarcolemma, the sarcomeres, the thick and the thin filaments, the myosin head, the actin filaments, so on. Now, as I've been saying all along, muscles don't just start contracting whenever they want to. They only contract when somebody tells them to. And who tells them to contract? Nerve impulse. So now it's time to start looking at the other side of muscle contraction, the nerve side of the contraction. And that's where we're going now. So muscle contraction can be broadly viewed as consisting of two phases. An electrical signal, the muscle action potential, moves along the sarcolemma, and by causing the release of calcium from the terminal cisterns of the SR, the electrical signal leads to a mechanical force of muscle contraction. That's referred to as excitation-contraction coupling. This really isn't a big deal to me. It's just a way of summarizing that, there are two main parts. There's the electrical side, and then there's the mechanical side. That's all that's going on here. But the term EC coupling, or excitation-contraction coupling, gets used a lot, shows up in the books a lot, might show up in other sources you have. So it was worthwhile covering it. And as long as you understand that there's the electrical part and then the mechanical part, I think we're good. So calcium. Remember, the muscle can't contract until the myosin head can actually grab that actin filament. And it can't do that when the muscle's at rest because troponin tropomyosin was in the way. It took calcium to move it out of the way. So calcium is the key to the whole thing working. Calcium, remember, was stored in the terminal cisterns of the sarcoplasmic reticulum, which you see in the illustration. Look in the far upper left. There we see the SR going around the outside of the myofibril. Then we're going to zoom in on the sarcoplasmic reticulum, and then the edges of the sarcoplasmic reticulum, the ones that ran right up alongside the T-tubule, remember those were called terminal cisterns, and those had little gates, little doors, like little garage doors. And it's the electrical signal that opens the little garage doors in the terminal cisterns, and that's what lets the calcium out. Normally those garage doors are closed, all right? And that's why muscles are not normally contracting. It's not until the garage door opens and the calcium comes out, drinking a beer while I'm doing this, that's when the calcium can then actually move troponin tropomyosin out of the way, and the myosin head can grab hold of that actin filament. When a muscle action potential propagates, a cool word, we're going to start using that a lot when we get to the nervous system, along the sarcolemma and through the T-tubules, it causes the calcium release channels to open. That's their official name, the calcium release channels. It's the muscle action potential that traveled down the T-tubules, ran alongside the terminal cisterns of the sarcoplasmic reticulum, opened the little garage door, and now calcium comes out. Calcium then flows all through the sarcoplasm, all around the actin and myosin filaments, and it starts moving that troponin tropomyosin out of the way, and now that myosin head can go ahead and grab that actin molecule on the thin filament. So, calcium binds to troponin tropomyosin, moves it away, myosin binding sites, okay. So you can see a kind of a cross-section view here in the illustration. But now, think about this. Let's say we have the nerve impulse, the muscle action potential, traveling along the sarcolemma, going down the T-tubules, running alongside the terminal cisterns, causing the calcium release channels to open. Calcium flows into the sarcomere. Remember, as long as calcium is there, that muscle will continue to contract because the troponin tropomyosin complex will keep getting moved away from the myosin binding site on the actin. And so once we start contracting a muscle, how the heck do we ever get it to stop? Seems like what we've learned so far, once you contracted a muscle the first time, it would stay that way for the rest of your damn life. And that won't be helpful because you will just be stuck there all contracted up. So how is that going to work? Think about that, okay? Once these binding sites are free, myosin free to bind, The calcium active transport pumps will pump the calcium back into the terminal cisterns, and that's how we are able to get muscles to stop contracting. Now the thing is, those calcium pumps, the calcium active transport pumps, are working 24/7, 365. So the moment the muscle action potential opens those little garage doors, once it opens the calcium release channels and the calcium flows out, those pumps are already at work pumping it back in again. And that's actually the way you want it to be. When you move a muscle, you want it to move only for the very moment you're thinking about moving it. You don't want there to be like a delayed reaction time where once you want a muscle to stop moving, it keeps moving for a while. That's not going to do any good. So those calcium active transport pumps, that's what makes muscle contraction very short term, lasts only moments, because those pumps are so good. They are pumping all that calcium back into the terminal cisterns right from the get-go, and that's why muscle contraction only lasts for a fraction of a second. What would happen if there were no calcium active transport pumps? Well, the calcium would stay in the sarcomere and the sarcoplasm, floating all around the actin and myosin molecules. It would keep moving troponin and tropomyosin out of the way. Myosin would keep binding to actin, and the muscle would stay contracted for the rest of your damn life. That's what would happen. So no calcium active transport pumps, that would be a disaster. You would never be able to stop the muscle from contracting. Let's take a peek here at the NMJ. The NMJ is a huge topic. It's really our introduction to the nervous system. This is our first look at the nervous system, which is the topic in the third section of the class, and it is by far the most important topic in all of Bio 201. So here we go, our first look. The synapse between a somatic motor neuron and a skeletal muscle fiber is the neuromuscular junction, the NMJ. Now there are several words in that sentence that you may never have seen before. The synapse basically means a junction, is what it means. It means where a nerve comes together with something else. Okay? So look in the upper left, you see it says the axon collateral of a somatic motor neuron. So neurons are the basic cells of the nervous system. We have both motor neurons and sensory neurons, and other neurons called interneurons. We'll get to all that later. Sensory neurons carry sensory information from your body to your brain. Motor neurons carry information from your brain out to your body. Motor neurons are what make things happen. Motor neurons in particular are what make muscles contract. Somatic means going to the body. When we get to the nervous system, we'll see that there's both the somatic nervous system and the autonomic nervous system. Autonomic nervous system takes care of all the unconscious things going on, like digestion and breathing and heart beating and stuff like that. Somatic nervous system is for voluntary movements of skeletal muscles, for the most part. So a somatic motor neuron is therefore the type of neuron that goes to a muscle to make that muscle move. And you can see here we have the axon, that's just part of the neuron. The axon terminal, that's the end of the neuron. And then at the very end, there's something you can see in the upper left again called the synaptic end bulb. And there are lots of names for this. Sometimes it's called the synaptic knob because it looks kind of like a doorknob. Sometimes it's called the terminal button because it looks kind of like a button. In French, it's the terminal bouton. And you have to say that in a way that feels superior to other people. It's the terminal bouton, English big dog. So lots of different names for it. Synaptic end bulb, synaptic knob, terminal button. You just have to get used to them. But the terminal button is the part of the nerve, the neuron. Nerves are made up of neurons. So it's the part of the neuron that comes into contact with the muscle. So that is the neuromuscular junction, all right? Where a somatic motor neuron comes together with a skeletal muscle fiber. That's what's going to let the electrical signal take place on the muscle. So at the end of the motor neuron, the axon terminal forms end bulbs where neurotransmitters are stored. Neurotransmitters are chemicals. And if you watch TV, you probably know the names of these. They're in movies all the time. Dopamine, serotonin, epinephrine is one. Right? There are lots of neurotransmitters. I'm going to make you learn somewhere around, I don't know, between 10 and 15, something like that. They have different effects in the human body. This is our first glance at one. So the neurotransmitter at the neuromuscular junction is acetylcholine. It's found other places in the body as well, but this is our first neurotransmitter. This is the neurochemical that's going to make muscle contraction happen. And it's stored in little packages called synaptic vesicles. They're basically like little plastic bags. Little plastic bags full of chemicals. The chemical is the neurotransmitter, specifically in this case acetylcholine, and the little plastic bags full like little sandwich bags full of acetylcholine are called synaptic vesicles. Exocytosis of acetylcholine occurs in response to a nerve action potential. Acetylcholine diffuses across the synaptic cleft to the sarcolemma. So some of those words you've seen before, some of them you haven't. Exocytosis, exo means out, cyto cell. Exocytosis means things leaving the cell. So when acetylcholine leaves the synaptic knob, that's called exocytosis. And there's a little space in between the synaptic knob and the sarcolemma. Remember, that's the cell membrane of the muscle fiber, of the muscle cell. That little space is called the synaptic cleft. So what happens is the nerve action potential. We saw the muscle action potential before. That's the electricity on the sarcolemma, on the muscle. The nerve action potential is the electricity that runs down the nerve. So the nerve action potential is what's going to cause the vesicles, the little sacs, to burst open, and they'll release the acetylcholine. So in the lower right, you can see that happening. The acetylcholine is released. It diffuses across the synaptic cleft down to the sarcolemma, and that's what's going to ultimately make the muscle contract. Let's talk about the events at the neuromuscular junction. You see a few different pictures here you can reference as we talk about it. So on the sarcolemma there are receptors for acetylcholine. Neurotransmitters generally work that way. Almost all of them do. There are a few that don't need them, but virtually all neurotransmitters and all hormones as well need receptors. The chemical has to have something to receive it in order to create an effect. And so notice how the sarcolemma in the upper left, see how the sarcolemma has these little dipsy doodles in it? Those are called junctional folds. So those little folded crevices, those little recesses, the junctional folds, that's where the acetylcholine receptors are. And that whole area, the place where there are all the junctional folds of the sarcoplasmic of the sarcolemma, we call that whole area the motor end plate. The MEP it's often abbreviated as. I'll try not to do that because you've already got a lot of abbreviations. But I would want you to know just basically that the motor end plate is where there are all those junctional folds, and that's where all the acetylcholine receptors are. And that's where the acetylcholine is going to bind. That's the word we use. The neurotransmitter binds to the receptor, and what does that do? Well, as it turns out, the receptor is actually an ion channel. In this case, it's actually a cation channel. Remember cations? That's not cation. That's cation. Cations are positive ions. And in this case, that receptor is actually a cation channel. And what are the cations involved? Sodium and potassium. Sodium is going to flow into the muscle cell, and potassium will flow out. As it turns out, the potassium flowing out is not a big deal for our purposes. Right now, what we want to focus on is the sodium flowing in. And when the sodium flows in, more of it flows in than the amount of potassium flowing out, that does what we call depolarize the membrane. Depolarize means it changes the electrical charge of the membrane. All right? You should already be thinking about this. Changing the electrical charge of the membrane, of the sarcolemma. So we are changing the electricity of the sarcolemma here. Keep thinking about that. So the flow of cations causes a depolarization called the muscle action potential. And now we've seen it. We've basically connected the two. We have made a linkage right now of everything we talked about before, the whole mechanical side of the contraction, we've now linked it with the electrical side. And we're going to review the whole thing in just a moment. You should already be in your head trying to put all of that together, because we've covered the whole thing now. So the muscle action potential travels down the T tubules, leading to a release of calcium from the sarcoplasmic reticulum, all right? Then, here's the deal. Remember how the muscle would stay contracted forever if that calcium stayed in the sarcoplasm? We had to get that out of there in order to make the muscle stop contracting. Well, once we release acetylcholine, as it turns out, it will bind to the receptor, so you can see it in the upper left and in the lower right as well. The acetylcholine will bind to the receptor, and the receptor will open. Remember, the receptor is actually just an ion channel. But what happens then is that the receptor lets go of the acetylcholine, and then the acetylcholine just floats around for a while. And then the acetylcholine can bind again. And that can go on and on and on and on and on forever. So once we release acetylcholine into the synaptic cleft, your muscle will now contract for the rest of your damn life if somebody doesn't get that acetylcholine out of the synaptic cleft. And who does that? One of the most powerful enzymes in the human body, acetylcholinesterase. And this is a big deal because there are drugs and toxins that work on acetylcholinesterase. So they will have a direct effect on muscle contraction, okay? So what would happen if acetylcholine were not hydrolyzed by acetylcholinesterase? That acetylcholine would stay out there in the synaptic cleft. It would keep binding to the receptors. The receptors, being the ion channels, would keep opening. Sodium would keep flowing through. It would keep creating muscle action potentials, which would flow along the sarcolemma. That would keep going down the T tubules. It would flow along the terminal cisterns. It would keep opening the calcium release channels. The calcium would keep flowing out into the sarcoplasm. It would keep moving the troponin and tropomyosin complex out of the way. The myosin head would keep binding to the actin filament. It would keep causing the power stroke, and the muscle would contract for the rest of your damn life. Whew. Did you follow all that? We know every bit of that. We've now learned every piece of that, and I expect you to know every piece of that, all right? That's what would happen. So here are just some pictures to show that I ain't making this shit up. This is real, all right? There you see different artists' conceptions of what this would look like. See, in the upper right, you can see those nice junctional folds down there on the sarcolemma. You can see the thick and thin filaments down below. You see that nerve terminal. All right, the junctional folds, the synaptic cleft. You can see the neurotransmitters coming out and doing their job. And here an actual photomicrograph. Oh my golly, will you look at this? You can see those synaptic envelopes, those synaptic knobs, the axon terminals, the skeletal muscle fibers. Yeah, it's real. It's actually out there. And there's a great picture as well. You can see the junctional folds, all right? And those would be lined with acetylcholine receptors. And up on the top, you can see those synaptic vesicles, little plastic bags full of acetylcholine, and they will be releasing acetylcholine into the synaptic cleft. It'll be going down into the junctional folds, binding to the acetylcholine receptors. And here's a cartoon of the same thing. So I put several pictures in because sometimes, you know, there's something about one picture that you go, wait, what is that? I don't get it. But if you look at another picture, you go, oh, yeah, okay, now I get it. So that's why. I just, took illustrations from different places. Maybe one will resonate better with you than another one will, okay? So, you have a handout this time called The Story of Muscle Contraction. And on your second exam, where are we? What year is this? Oh, yeah, 2020. So on your second exam, I'm going to have a question that says, tell me the story of muscle contraction. And that's all you have to do. You just have to tell me the story of muscle contraction. And so here it is. Here is the story of muscle contraction. Let's review it one more time. A nerve action potential causes the release of acetylcholine. Acetylcholine binds to postsynaptic receptors, opening sodium channels on the sarcolemma. The influx of sodium ions causes the muscle action potential. Acetylcholinesterase has to hydrolyze ACH, otherwise the muscle will keep contracting forever. The muscle action potential opens the calcium release channels in the terminal cisterns of the sarcoplasmic reticulum. Calcium floods the sarcoplasm. Calcium binds to the troponin tropomyosin complex, exposing the myosin binding site on the actin. Myosin heads bind to actin. That causes the power stroke. That rows the thin filaments past the thick filaments, and the muscle contracts. Then those calcium release channels have to pump all that calcium back in to the terminal cisterns of the sarcoplasmic reticulum, otherwise the muscle would contract forever. Then the troponin tropomyosin complex slides back into place, once again blocking the myosin binding sites, and the muscle stops contracting. There you go. We now know the entire story of how a muscle contracts.